everyone, welcome back again. So as we all know that DNA damage and mutation will cause the change in the nucleotide sequences of the DNA. But how this damage is actually repaired? So it is because of the mechanism that is what called as DNA repair mechanisms. So the topic for today's video will be DNA repair mechanism, where we are going to see the direct reversal pathway, what are the mechanisms behind the single-stranded DNA damage and also double-stranded breaks. So let's start with the topic and understand each and every pathway that is required to repair such kind of DNA damage. So the first pathway over here is direct reversal. And if you see direct reversal mechanism, that is further classified into two. That is photoreactivation mechanism and the other is methyl group removal. Now what do you understand by photoreactivation? Photoreactivation is a pathway that is used to correct any kind of UV mediated damage. So this pathway is to correct any kind of UV mediated damage, and we know this UV damage is because of the UV light and which causes the formation of thymidine dimer. So this photoreactivation mechanism, it is used to correct the thymidine dimer that is formed because of this UV rays. So it is used to correct pyrimidine dimers or thymidine dimer. This is a light dependent repair. As you can see in the name itself, it suggests photoreactivation, means in the presence of photon on light, something will be activated. So what will be activated? Enzyme will be activated to repair this damage. And this is a single step process and it requires an enzyme that is going to repair this thymidine dimer ka damage. So the E. coli enzyme, that is DNA photolyase, which requires a 500 nanometers of light. So it requires a visible spectrum that is 500 nanometers of light, and this 500 nanometers of light is a blue wavelength of light. And we know visible spectrum is between 400 to 700 nanometers, but this is going to absorb 500 nanometers, means that is blue light. That is why this is called as photoreactivation, means in the presence of light, something is stimulated or activated. So here the enzyme is stimulated by requiring this 500 nanometers of light. So now we will see the mechanism that is involved to correct this type of damage. So you can see in the figure as well, this is double T. Double T means thymine. That pairs with A. So thymine actually pairs with A. So what happens here? In the presence of UV light, this will thymine that are present adjacent to each other, that will get paired up. And now you can see a triangle, that is black triangle in the next step. That is thymidine dimer in the presence of UV light. But how this is actually corrected? This is because of the DNA photolyase enzyme that is used to absorb some kind of light, that is 500 nanometers, and it used to lyse and split the thymidine dimers form. That is hydrogen bonds in between the thymidine dimer. So you can see this green color is a DNA photolyase enzyme, and utilizing this visible light, that is nanometers of light, it will get activated and it will be dissociated once the thymidine dimer damage is resolved. Now you can see this is the original form as we are seeing here, and in this way this damage is resolved by DNA photolyase enzyme, and this is what called as photoreactivation. But why this is called as direct reversal? Because this is just a single step pathway. Now we will see the next mechanism that is included under direct reversal as well, and this is called as methyl group removal. Means this pathway is used to correct such damage in which methyl group is added. So it is used to remove the methyl group that has been added to the nucleotide sequence. So for what it is used? It is used to repair O6-methylguanine. And we know if guanine is methylated at 6th position, it will form O6-methylguanine, that is a base analog. But this base analog is not the nitrogenous base that is incorporated in the DNA. So this methyl group has to be removed. So this will be done by this mechanism. And what is the enzyme? The enzyme will be specific to this base analog only. It will be O6-methylguanine DNA methyltransferase. Means to this specific enzyme will be activated, and the name is DNA, that is it is DNA dependent. It will bind to DNA that will transfer the methyl group from guanine, and ultimately this O6-methylguanine will be converted to its original form, or you can say guanine. So it catalyzes the conversion of this base analog to guanine itself. Now you can see in the structure as well, this is O6-methylguanine nucleotide. Means the methyl group is added to the 6th position. This is 1, 2, 3, 4, 5, 6. 6th position, the CH3 group is added. And once the CH3 group is added, this will be recognized by a specific set of enzyme that is called as DNA methyltransferase. And this DNA methyltransferase is activated because it contains cysteine residue in its active pocket. You can see here, it contains cysteine residue in its active pocket, which is an amino acid. And once this activated form acts on this methylated guanine, it becomes inactive, because you can see this CH3 group from guanine is now added to its active pocket, and hence it becomes inactive. And that's why this is called as methyl group removal, because the enzyme that is methyltransferase, it is transferring that methyl group from guanine to its active pocket itself, and is transforming O6-methyl guanine to the normal nitrogenous base, that is guanine, and hence the damage is repaired. So this is all about the direct reversal mechanism, where we have seen two pathways, that is photoreactivation as well as methyl group removal. Now we are coming to the single strand DNA damage. What happens if one strand of the DNA is damaged? So that is what called as single stranded DNA damage. So under this we have to study three mechanism, or you can say three pathway. What are these three pathways? The first is BER, that is base excision repair. The second is nucleotide excision repair, and the third is mismatch repair. So firstly we are going to see what is BER. The name itself suggests SR repair pathway that is going to excise base and repair this damaged nucleotide. So here what will happen? This is going to excise the base only. So now let's see the pathways. So this is used to correct the oxidative damage if it has happened to the cell, if any kind of hydrolysis event has occurred, or if any alkylating agent has acted upon the DNA and they have added any kind of alkyl or ethyl group. So if these groups are added, or if these damages are occurred in the DNA, that will be removed by this base excision repair. So what are the requirements of this repair pathway? So the requirement is the enzyme, that is DNA glycosylase enzyme, that is going to break the N-glycosidic bond. As the name itself suggests, glycosylase. Lase means it is going to split something. What? Glycosidic bond. Which bond? There are two types of glycosidic, O and N-linked glycosidic bond. So this time it is going to break N-glycosidic bond. So the next enzyme is AP endonuclease. The third requirement is phosphodiesterase enzyme. DNA polymerase 1 is needed, as well as DNA ligase enzyme. So now we'll see the entire pathway, how these enzymes are utilized here. So coming to the base repair pathway, so you can see in the diagram as well. So here I have mentioned this is used to correct the oxidative damage, hydrolysis event, as well as alkylating, any kind of alkylation reaction if it has happened. And also it is used to correct the deamination part. If any nitrogenous bases are deaminated, so it is going to correct this damage. So here the example, or you can say the diagram is given for this you can say deamination reaction. As you can see in the diagram as well, this diagram represents for the base excision repair, and near to this some deamination reaction is being shown. So here you can see, I'll just zoom this diagram. So you have to just see this. So here if you see, this is a cytosine nitrogenous base. So when it is deaminated, it will form uracil. And if 5-methyl cytosine is deaminated, it will form thymine. So here we are going to discuss the first one. When cytosine is deaminated, it will form uracil. So now let's come back to the diagram and discuss this. So here you see the duplex with UG mismatch. Why UG mismatch? Because you can see G is there, but instead of C, here comes U. Matlab C should be there, but you can see a blue bar, means U has been incorporated. Why? Because cytosine was there, and that cytosine is deaminated into uracil, and now uracil is there. So this pathway is also used to correct the 4th type of damage, which is called as deamination reaction. So if there is any kind of deamination event, so that will be corrected by BER mechanism. So duplex with UG mismatch is not there, UNG mismatch. That will be recognized by the very first enzyme, that is called as uracil DNA glycosylase. And why uracil DNA glycosylase is activated? Because this is specific to uracil, and it is going to cleave all the bonds, you can say glycosidic bond, and releasing that uracil base. And you all know that if you see the structure of DNA, this is a deoxyribose sugar, this is a base, and this will be the phosphate group. And how this phosphate group and deoxyribose sugar is attached? It is because of the phosphodiester bond. And what about this base? This base is attached to this sugar by a glycosidic bond, and this will be N-glycosidic bond. So what happens here? Once this enzyme is activated, it will break or it will cleave this glycosidic bond. And once this glycosidic bond is cleaved, it will leave a blank space. Why? Because the base is removed. Still the phosphodiester bond is there. So this site is called as AP site, that is apurinic or apyrimidinic site, that is without any base, and still a phosphodiester bond is there. So you can see a blue bar is still there, which represents a phosphodiester bond is still there, but only U is removed, means it will leave a blank space, that is called as AP site. Apurinic or apyrimidinic site, but here it is uracil, that is a pyrimidine, so it will be apyrimidinic site. And this site will be recognized by a second set of enzyme that is called as AP endonuclease. So AP endonuclease will be specific to AP site. Now what's the function of endonuclease? It will cleave at the internal segment. If it will create a nick in the phosphodiester bond, you can see the nick is created and phosphodiesterase will be activated, that will remove this phosphodiester linkage, and ultimately there will be a nick now. Now that will be sealed by DNA polymerase 1, as well as the gaps will be filled by DNA ligase enzyme, and ultimately the mismatch is repaired. So which DNA polymerase will be filling this gap? That will be done by DNA polymerase 1, because we know DNA polymerase 1 in prokaryotes or E. coli, it is known for DNA repair and gap filling. So you can see we are starting DNA repair pathway, and it will fill the gap. Ultimately it will be sealed by DNA ligase, that will be catalyzing the last phosphodiester bond formation. And now you can see once the DNA is repaired, there is no change in the helix conformation. As you can see, there is no helicase enzyme action, there is no change in the helix conformation, as only base is removed. So this type of repair is also called as non-helix distorting repair system. So this is about BER. Now let's come to the next type of pathway, that is NER, nucleotide excision repair. So if you see this pathway, this is going to repair UV light chemicals. And just now we have seen this UV light mediated damage, it is corrected by DNA photolyase enzyme. But this photolyase enzyme, it is not ubiquitous for all the cells. It is not present in all the cells. What if this enzyme is not present? So that the thymidine dimer formation, that is corrected by, or this damage you can say it is repaired by NER mechanism. So again here we are going to see the UV light chemicals mediated damage, that is going to be corrected by NER mechanism itself. So it corrects UV light chemical damage. The requirements for this is UVR ABC complex, which is called as axi-nuclease, helicase 2, which works as UVRD, DNA polymerase, and ultimately the gaps will be sealed by DNA ligase enzyme. So now here we will see the diagram and what is the pathway, or you can say the repair mechanism for this NER pathway. So you can see UVR ABC complex that is needed to repair this. So suppose if there is thymidine dimer formation and if there is adjacent thymine ka pairing. So this will be recognized by a UVR ABC complex, and out of this UVR A and B are responsible for recognizing this thymidine dimer. As you can see in the diagram as well, this is UVR AB complex. B is a monomeric subunit, as well as you can see the green part, that is double A, means it is a dimeric component of UVR APC complex. So this way is going to recognize the damage. Once the damage is recognized, the A part is dissociated. Why it is dissociated? Because it has recognized that damage. Now it will recruit and it will send a signal to UVRC. Now once UVRBC are recruited to the damaged site, they will act as a exonuclease enzyme and they will make a cut towards the 5' end and also towards the 3' end. So two cuts will be made near to the damage. So you can see a thymidine damage here, or you can see the thymidine dimer formation. To this 5' cut is also made, and also 3' cut is made. 5' cut is made to the 5' end and to this 8th phosphodiester bond. So towards the 5' end, 8th phosphodiester bond is cleaved, and towards the 3' end, 4th and 5th phosphodiester bond is cleaved. Now once this bond is cleaved, means they are exonuclease. So this stretch has to be removed, because this is a strong long stretch you are seeing, and this stretch has to be removed because the nicks are only created, and now this stretch will be removed by a separate set of enzyme that is called as UVRD. And this UVRD here, it is a helicase helicase 2. It is an helicase enzyme that requires ATP, and it will unwind the DNA duplex and it will release that nicked strand, and ultimately that part is removed. As you can see in the next diagram, here there is a gap, and this gap will be filled by a DNA polymerase 1 enzyme, and ultimately the sealing will be done by DNA ligase. So this is all about the NER mechanism, and in this mechanism you have seen the helicase enzyme action, means it is opening the DL helix, means at some point of time it is going to distort the conformation of DNA helix, or the conformation is changed. That is why this type of repair pathway is also called as helix distorting base repair mechanism that we have not seen in base repair pathway. So here you have to just remember, if helicase will act, it is going to distort the helix conformation. So what are the defects that are related with NER mechanism? So there are two defects that you have to recall and remember. The first is xeroderma pigmentosum, that is an autosomal recessive disorder. So if there will be a defect in NER mechanism, there will be no correction of this thymidine dimer formation, and it will lead to xeroderma pigmentosum at some point of time, which is a kind of cancer, you can say. And you have to recall, it is the autosomal recessive disorder, and the other one is Cockayne syndrome. So this is all about NER mechanism. So now let's come to the third type of mechanism, that is mismatch repair. So if you see mismatch repair, this is going to correct the replication error. So what do you understand by replication error? Error that has just occurred after the replication process. So suppose if the replication process is completed and if there is any kind of mismatch, so that will be corrected during the mismatch repair mechanism. So let's see here, the requirements for mismatch repair is the mute HLS complex. The second is DNA polymerase 3, and the third is DNA ligase enzyme. You can see DNA polymerase 1, third. Why DNA polymerase 3 is needed here and every time it is 1? Because it is used to correct the replication error. And we all know that DNA polymerase 3 is the main replicating enzyme that is used for leading as well as lagging strand synthesis. So it is corrected just after replication. So this polymerase is activated and DNA pol 3 will only correct this replication error, and it is involved in mismatch repair pathway. Now this mute H, L and S, there are three different enzymes, so it is a heterotrimeric complex because three different subunits are present. So mute H has an endonuclease action which recognizes the Gartach sequence. The second is mediating the interaction between HS and S. And what is the function of S? It is going to recognize the damaged site. Now here we are going to see the full detail and you can see the full process, how this repair takes place. So you can see this is a double-stranded DNA. You can see this is the parental molecule. So once the parental molecule is formed, or that parental molecule will be replicated, and to this replication daughter strands will be formed. But sometimes this daughter strand, some incorrect base will be added. So how this base is removed? It is because of MMR repair mechanism. So you all know that how the replication starts in E. coli. It starts from the Gartach sites. This is origin of replication which is recognized by DNA A enzyme. And you all know that how this parental strand is working as a parental strand for the daughter template. Why? Because this adenine residue has methyl group at its 6th position. So once the parental molecule is replicated, daughter strand will be also formed. So daughter strand will also have Gartach site. But this time this Gartach site will not be methylated. Why? Because this methylation is done by a separate set of enzyme. It is not because of replication this methyl group is added. This is because of the separate group of enzyme that is called as DNA adenine methyl transferase that will transfer a methyl group to this adenine residue. And now this parental molecule is a mature parental that will go for round of replication. But if these are replicated and the daughter strand is formed, means they will not have any methyl group. But if incorrect base is added, so after the replication, DNA replication is completed and before the addition of methyl group, this damage is repaired. So if there is any kind of mismatch, that will be repaired in this window, that is after the DNA replication is completed and before the methyl group addition. And once this damage is repaired or it is corrected by the DNA polymerase enzyme, and ultimately there will be a methyl group addition, and now this time the daughter template is also become the mature template and it will go for next round of replication. So here we will see the pathway. You can see this is a double-stranded template, and this mute H enzyme which is recognizing the GATAC site. So it is recognizing the GATAC site because this GATAC site is non-methylated in the outer strand. And you can see mute S which is recognizing the damage, and this yellow one is mute L. So once the mute H binds to the GATAC site, so it will recognize the origin site, and this mute S is binding to the mismatch. As you can see, all are black in color, that is a bonding, you can say the hydrogen bonding between nitrogenous bases, but here you can see blue and red part, that is an incorrect base pairing. So mute S will bind to the damage site. As you can see the function of mute L, that will bind to the damage site. Once they will bind, mute L will mediate the interaction between mute H and S. You can see the DNA is now wrapped, it is folded because of mute L. And now mute has an endonuclease action that will cut the daughter strand where there is an incorrect base, that will cut just after the GATAC sequence till the damage site, and that damage you can see it is now empty. This area is now empty. That will be filled by DNA polymerase third enzyme, and the ultimate gaps will be sealed by DNA ligase. And you can again see this time this mute H is endonuclease, so it is going to just create a nick. But how this strand is removed and the gap is created? This is because of the helicase enzyme that has been activated after this, and ultimately helicase is going to unwind the DNA helix and it is going to remove that nicked part, and ultimately the stretch will be released. But this requires a helicase. So again, it is a type of helix distorting repair pathway that is going to distort or change the conformation of DNA duplex. So this is all about the single-stranded DNA damage. If there will be a strand, any kind of damage in the single strand, that will be repaired by BER mechanism, NER repair mechanism, as well as MMR pathway. Now let's come to the last type of repair mechanism, that is double-stranded break. Suppose if there is any kind of double-stranded break in the DNA, so that will be repaired by a double strand break mechanism. And again here you are going to study two pathways here, that is non-homologous end joining and the one is homologous end joining. So firstly we are going to see non-homologous end joining. So it is used to correct any kind of ionizing radiation as well as X-ray damage. So you know that ionizing radiation ka example is X-ray, beta ray, gamma ray. X-ray, alpha, beta, gamma. So all these rays comes under ionizing radiation, and whenever they act on the DNA, they are always going to generate the overhangs. They are always going to generate the streaky end. So what is streaky end? Where you can see the overhangs that are formed, that is called as streaky ends or staggered ends. So once the streaky ends is formed, that is required, the NHEJ machinery is required. So this sticky ends that is generated, you can see both the strands are affected here. Means it is going to be recognized by a separate site of enzyme that is called as Cu7080 heterodimer. So the very first enzyme that is going to act here is Cu proteins. So here Cu will be of a 70 kilodaltons weight and it will be of a 80 kilodaltons molecular weight. So both these molecular weight Cu proteins, they will form a heterodimer, and you can see here to both the strands they are binding. Means heterodimer is binding to both the strands. Once this heterodimer is bind, it will send a signal to DNA dependent protein kinase enzyme. So you can see DNA dependent protein kinase enzyme, it is binding to both the Cu proteins, that is Cu80. And DNA protein kinase enzyme has one main activity that is called as autophosphorylation. Means it has the capacity to autophosphorylate itself. Now what it will do? It will autophosphorylate itself. You can see it is adding the phosphate group onto itself, and means DNA protein kinase enzyme is now activated. Once it is activated, it will activate its substrate. You can see protein kinase enzyme means it is going to add phosphate group on its substrate. And what is the target for this? It is Artemis enzyme. And now the Artemis enzyme will be activated. That will cleave the overhangs. You can see that will cleave the overhangs. So this overhangs will be cleaved by Artemis and ultimately will get this blunt end kind of structure. So previously it is shown blunt end, but it's not like that. It is previously staggered hand, and this blunt has been created by this Artemis, which is going to cleave this overhang flanking region, and ultimately the gaps will be sealed by DNA ligase enzyme. So here you can see ultimately DNA ligase enzyme will act, which will be in the form of XRCC. So this DNA ligase machinery will be XRCC, DNA ligase 4 enzyme, or along with AXLF. So this is just a complex of DNA ligase enzyme that will cause the pulling of the strands towards each other, and ultimately there will be a sealing of phosphodiester bond, and this is what called as non-homologous end joining, where the ends will be joined but they are non-homologous to each other, means both the strands are different in nature, they are non-homologous to each other. So this is about the non-homologous end joining mechanism. Coming to the last mechanism of today's lecture, or that is the last mechanism of double strand break, that is homologous end joining. Means here homologous region is used to repair this damage. So let's see this mechanism in detail. So here again it is used to correct the ionizing radiation. So this double strand breaks, they are always created by ionizing radiation, that includes X-ray, that includes beta ray, that includes gamma rays. So this will be corrected by the homologous end joining this time, and the machinery will be homologous end joining machinery. As you can see, there is a double strand break here. You can see there is a double strand break here, and again this time it will be a staggered cut. It will be a staggered cut. Nick will be followed by the and resection. This time N complex will be activated, that is mRN. This time the mRN complex will be activated, that is going to recognize this damage. We have seen previously it is Cu7080 heterodimer, but for HEJ it will be mRN. M stands for MIRI11, R stands for RAD50, and N stands for NBS1. So it stands for NBS1, RAD50, and that is MIRI11. That is MIRI11. So all these enzymes will work for the end resection. And resection means to split something. So they are going to split the ions. They are going to remove the nucleotide bases from the end. That is called as end resection. So there we have seen Artemis was performing this function, but here this complex itself will recognize this damage and will perform the end resection. Once the end resections are done, 5' and is cleaved. Now once the 5' and is cleaved, that will leave the 3' overhang. Suppose if 5' part is cleaved, that is going to leave the 3' overhang. Now the 3' overhang will be flanking. That will be maintained by a replication protein A enzyme, and we all know that this replication protein A enzyme is a single stranded DNA binding protein which is going to maintain a single strand in a or any kind of strand in a single stranded fashion. So this RPA is going to maintain a single strand state, so that both the DNA helix or you can say both the strands should not get coiled up, and again they should not come in a duplex form. And again this RPA, now the next step what will happen, this RPA will be replaced by BRCA2. You can see this is a tumor suppressor gene. So this RPA will be replaced by BRCA2, and what's the function of BRCA2? Similar to RPA. So RPA will be just replaced by this BRCA2. Why? Because now the next enzyme will be acting, that is called as RAD51. This RAD51 needs BRCA2 for its function. So once RAD51 is recruited, it will add BRCA2 to this template and it will replace RPA, and this function, now this is single stranded maintenance, it will be performed by BRCA2 itself, which is a type of tumor suppressor gene that is associated to breast cancer. Now this RAD51 is activated, which is very important as it performs the phenomena of a strand invasion. Why this strand invasion is performed? Because you can see the homologous sequences. Now this and is cleaved means 3-OH groups are free. Now they will cause the phenomena of strand invasion, and ultimately you know about homologous recombination, there will be a D loop formation, and ultimately the damage will be repaired. So this type of damage is called as homology directed repair, because it utilizes the homologous chromosome, and the strand invasion is performed by RAD51 protein. So this is all about the DNA repair mechanism, where we have seen what is the direct reversal, we have seen the photoreactivation pathway, and also we have seen the methyl group removal. We have seen the single stranded DNA damage. In that we have seen 3 pathway, BER, NER as well as MMR, and also we have seen double stranded break repair, that is NHEJ and also HEJ. So all these topics are important for gate biotechnology point of view. So here we are going to revise for gate biotechnology. So if you want to revise more topics in a similar way, so you can just write your comments, or you can just write your topics in the comment box. We will come up with that video soon. So thank you for watching this video. If you like the video, do not forget to like, share and subscribe to our channel, that is Biotecnika. So meet you in the next video. Until next time, this is your host Riya. Thank you everyone. Take care. Keep learning.